This is Matthew Armstrong here to deliver a methodology for adding Thixotropy to Oldroyd-8 family of viscoelastic models for the characterization of human blood. This is the outline. I'm going to go through some of the motivation and we'll talk a little bit about the complex character of human blood. We'll then dive into the models to include the modified enhanced thixotropic Horner-Armstrong-Wagner-Beris, and then we'll see about the Oldroyd-8 framework. And we'll talk about the fitting procedure that is identical for each of the models discussed today. And then we will determine from our model predictions and comparisons which model has the lowest cost function. This is human blood. It is a thixo-elasto-viscoplastic material. And moving from left to right, the thixotropy in this case refers to a microstructure which has a time of structure breakage and a time of structure rebuilding. And for us today, the structure is the rouleau, which are tiny coin-like stacks or coin stack-like structures that form in blood under low shear rates. At low shear rates, elasticity refers to the stretchiness of the rouleau. The viscosity refers to not only the resistance to flow of the plasma and individual red blood cells, but also the contribution to the overall stress from the viscosity of the rouleau. And then the plastic refers to the irreversible breakage of the rouleau as they undergo shear stress. And here is another cartoon to describe the rouleau. If you look in the upper right-hand corner, you can see those coin stacks of red blood cells we were talking about. And of course, blood being complex is shear thinning, which means that the viscosity changes as a function of the shear rate. It has a non-zero yield stress, which is the minimum stress required to induce flow. It does has viscoelasticity and plasticity. And in fact, the viscoelasticity has a contribution from the plasma and red blood cell backbone and also the rouleau. And the thixotropy refers directly to the rouleau. And during different flow conditions, there's going to be forming and breakage of said rouleau. The basis of our experimental protocol is the steady state experiment, steady state where we vary the shear rate and measure the stress. And we are going to be fitting a certain number of parameters to the steady state first. And what we're looking at here is an example of what the steady state experiment might look like. On this set of axes, I've also plotted on the right-hand side the structure between 0 and 1, where 1 is a fully formed structure and 0 is there is no structure because of the high shear rate. All of our models today have the feature of allowing us to look at the elastic contribution to the stress, which is also the green dashed line, and the total stress is black and the viscoelastic stress is the blue. These are the step down and step up experiments whereby we begin to fit the transient parameters to on the top part of the slide. This is the step down and shear rate experiment whereby we're at a high shear rate and we bring down the step down the shear rate to a lower value. Moving from left to right across the top, we can see what that does to the stress, right? Immediately we have a drop in the stress, but as the microstructure begins to reform, the stress starts to recover. And on the far right, upper right-hand corner, we can see the structure reforming. On the bottom portion of the slide, this is the opposite, the step-up experiment, where we're at a lower shear rate and we step up. And then we can see immediately what happens to the stress as we step up. We immediately start to break that structure. And so we have a peak in stress, and then the structure begins to dissipate and break apart. And again, in the lower right-hand corner, we see what happens to the structure during this step-up experiment. Our model fitting procedure is simply to fit each model to steady state, only the steady state parameters first. We're going to normalize the stress here because there's 4 orders of magnitude difference. Second step is to hold those steady state parameters constant and then fit to 4 step ups and 4 step downs. And then we're going to hold all of those parameters constant, and we're going to make a series of predictions of small amplitude, large amplitude, and unidirectional large amplitude oscillatory shear. And we've developed a code that will directly take the logarithmically spaced data from Trios and fit that directly using a global stochastic algorithm. At the bottom, we can see the formulation of our cost functions. On the far left is the steady state, then is the difference of perfect squares for steady state, and then we have the oscillatory shear and all the other transient flow condition cost functions. Lower right-hand corner shows the Akeki information criteria, which assesses a penalty for additional parameters, and this is how we're going to compare 3 Oldroyd-8 family of models and the base enhanced thixotropic mHAWB. This is a cartoon depicting the large amplitude oscillatory flow paradigm. On the left-hand side, we're showing 3-dimensional space with stress, strain, and shear rate. The louse itself is a sinusoidally oscillating function of sine. Taking the first time derivative gives us the shear rate. In the upper right and lower right side of the slide, we see the Lissajous-Bowditch projections. On the top is the viscous projection, whereby we plot stress versus shear rate, and on the bottom is stress versus strain. And this is a mapping. This is the mapping of how we can interpret the Lissajous-Bowditch curves. And the 2 equations depicted at the top are the small amplitude oscillatory shear reconstruction of stress using G prime and G double prime, which happen to be the first harmonic solution. And the transition into louse implies that we are now using the 3rd, 5th, 7th, and etc. harmonics, where souse by itself implies only the first harmonic to recover the solution. Note that the unidirectional louse is depicted at the bottom right-hand side, and the rheometer may speed up and slow down, but it never changes direction, which we think is more indicative of what we might expect to see in human blood. With respect to our models today, this is how we're going to model the thixotropy. It has a shear breakage term and 2 shear building terms. This is the enhanced thixotropic model. On the right-hand side is the viscoelastic contribution from the rouleau. And this particular framework that you're looking at right here will be applied to each of the models simultaneously. Of course, our basis model of comparison recently published, Horner et al., 2018 and 2019, based on a Maxwell equation with a White-Metzner modification using the cross to calculate the viscosity as a function of shear rate. This is well published, and I would, if I had any more questions about this, go to the references. All in all, we have 5 ODEs, 6 algebraic equations, 12 parameters. 9 of those parameters are getting fit to steady state, and the other 3 to the 4 step up and 4 step down experiments. Here's the result. On the left-hand side, you're looking at all the fits, starting at the top with the steady state, followed by the 4 step downs, and then the 4 step ups. On the right-hand side, we're starting to see the predictions using the mHAWB model with the donor from December 17th. Moving forward, here are the elastic and viscous projections of louse and UD louse respectively. And overall, we are capturing the qualitative features of the oscillatory shear. Here are the best fit parameter values for all 5 donors tested in this project. Notice that we have now been able to capture the average parameter values of human blood for using the mHAWB, and we also have an average value of the cost function for steady state and an average value for the cost function of the transient. Those 2 numbers are going to be the basis of comparison as we bring in the other Oldroyd-8 models. So this is the enhanced thixotropic Giesekus equation framework for the viscoelastic backbone of the blood plus the individual RBCs. And I would check the Bird-Armstrong-Hassiger dynamics of polymeric liquids for more information on these equations. Here are the fits on the left-hand side, and on the right-hand side are the predictions. And here are the predictions of the large amplitude oscillatory shear and unidirectional oscillatory shear experiments using the enhanced thixotropic Giesekus model. And here's the final roll-up of the cost functions of steady state and transient parameters. And at the top, we see the average values of all the parameters, rheological parameters. And we're going to follow the same procedure for the enhanced thixotropic Corotational Jeffries. Again, these equations came right out of Bird-Hassiger text. On the left-hand side, we see the fits, and on the right-hand side, we see the small amplitude oscillatory shear predictions. Here are the elastic and viscous projections of the louse and uni-louse respectively, and here are the average parameter values to include the average cost function of steady state and the transient fits. Okay, elastic model that we chose to incorporate was the enhanced thixotropic convected Maxwell. The equations are here, and again, they're in the Bird and Hassiger text. On the left-hand side are the fits, on the right-hand side are the small amplitude oscillatory shear predictions. Here's the louse and UD-louse predictions. Again, the average parameter values, cost function of steady state and transient. And what we can do at this point is, with our massive library of model parameters, rheological model parameters, we can start to look for statistically meaningful correlations between our model parameters and the physiological values of human blood over the 5 donors. We don't have time to show you those results today. Here's the model comparison. From left to right, we have mHAWB, Giesekus, Corotational Jeffries, and Convected Maxwell. Bottom line up front, mHAWB outperformed everything, and it had about somewhere in the middle for the number of parameters. At the very bottom, we show the average values of the cost function for the small amplitude, large amplitude, and UD-louse experiments. And then we roll it up ultimately with a total score, which is showing us that mHAWB came in first and Giesekus was second. Future work, we want to ask ourselves the question, how does aspirin affect those model parameters? And what does it do to the rheological fingerprint of human blood? What does it do to the mechanical contour mappings of the viscous and elastic projections? Also, we want to start marrying up those model parameters, those best fit model parameters, with statistically meaningful correlations with the physiological data from the lab. All of this work I just showed you was recently published in Physics of Fluid, and I'd like to thank our collaborators at University of Delaware, Dr. Wagner, Dr. Beris, Jeff Horner, who's now Dr. Horner, and the collaborators at United States Military Academy, including Major Corrigan, Jesse Trump, and Major Milner. Here's the cadets that actually took a lot of this data with me while down at UD Laos with Jeff Horner, and of course, Dr. Rogers. Thank you.